Hello, my name is Rani Rami, and today we will be discussing how accurate are the scientific findings of the news article, How Humans Lost Their Tails, to the primary article, The Genetic Basis of the Tail Loss Evolution in Humans and Apes. For my news story, I selected an article from the New York Times called How Humans Lost Their Tails. In this biology course, we discussed that evolution represents a shift in species features over numerous generations due to natural selection. The theory of evolution is founded on the assumption that every species are connected and constantly evolve. This newspaper is related to evolution because it discusses arguably the most significant anatomical evolutionary modifications due to lineage, ultimately to humans and apes, which is the disappearance of the tail. Recently, a group of experts in New York claimed to have identified the mutated gene that may have resulted in the abolition of our tail. According to a recent study published, when scientists introduced this genetic mutation in mice, the mice didn't develop tails. This significant structural shift had a substantial influence in our evolution. The tail muscles of our ancestors morphed into a hammock-like web over the pelvis. In this article, it discussed the findings on how tails have evolved into caucasus over the years. Among the most significant anatomical evolutionary alterations throughout the path leading to humans and apes is the disappearance of the tail. This structural reconfiguration in early humans was widely considered to have supported a unique locomotor technique and aided in the emergence of upright walking in humans. The exact genetic process that aided human tail loss evolution is still unexplained. But according to a group of scientists, the introduction of a single ALU element into the DNA of the human ancestor drove tail loss evolution. Exclusively primates have ALU elements, a form of jumping gene or transposable element, TE. They show that this ALU element, which is introduced into an intron of the TBXT gene, connects with a nearby ancestral ALU element expressed in the opposing chromosomal position, and this results in a human-specific unconventional splicing occurrence. The goal of the study was to investigate the impact of splicing occurrence, so scientists created a mouse model that expresses both full-length and exon-skipped variants of the mouse TBXT ortholog, stimulating the production of human TBXT products. They investigated the 31 human genes associated with tail formation, as well as their ape orthologs, in the expectation of finding a gene mutation linked to the disappearance of tail in humans. They looked at the protein structural conservation across human genomes and those of their closest ancestors, the Old World monkeys. Also, scientists searched for the human-specific genomic reconfiguration in the non-coding areas of gene associated with tail formation. In the findings, unexpectedly, a human-specific ALU element was discovered in intron 6 of the TBXT gene. TBXT is a substantially maintained transcription element required for establishing the mesoderm and ultimate endoderm throughout embryonic growth. In tailed species like a mouse, which has heterozygous abnormalities in the coding area of the TBXT orthologs, results in the loss or the decreased shape of their tail. At the same time, homozygous variants are often not viable. Also, this ALU addition belongs to the ALU-Y subgroup, a young, non-human specific subgroup found in the chromosomes of humans and Old World monkeys. whose activities correspond to a period when ancient humans lost their tail. Science has found evidence that intronic addition of any ALU-Y element drove tail loss evolution in humans. They hypothesized that this component engages with an adjacent ALU-S times one component in the adjacent intron, resulting in an alternate spliced variant that bypasses an adjacent exon rather than interrupting a splicing region. In conclusion, though the ALU-Y inclusion could have triggered tail loss evolution in humans, further genetic alterations could have stabilized the no-tail trait in primitive humans as shown in Figure 4. The ALU-Y addition in the TBXT was a pivotal genetic occurrence in the human shared ancestor tail loss transition. Subsequent genetic modifications could well have then stabilized the ancient human no-tail appearance. The particular evolutionary benefit of losing the tail is unknown. However, it is probably involved in improvement in the non-arboreal living. Nevertheless, given the disappearance of the tail could have entailed an evolutionary trade-off of neural tube anomalies as evidenced by the existence of spinal cord deformality, they can presume that the selection benefit was reasonably high. Figure 4 is a representation of the progression of tail reduction in ancient hominoid species. Is this news article accurate? Yes. It accurately covers the findings of the primary article. The news article presents the results in a simplified and narrative format, which may aid the reader's comprehension of the primary article. Let's look at the evidence. The scientists and the study's objectives are introduced in the first section of the news article, where they said, now a team of scientists in New York say they have pinpointed the genetic mutation that may have erased our tails. When the scientists made this genetic tweak in mice, the animals didn't grow tails. This corresponds to the main article in which a scientist professed to have created a mouse model that expresses the mouse TBXT ortholog, stimulating the depth of human TBXT product. The primary article states, we found that mice with a genotype exhibit the complete absence of a tail or shortened tail, supporting the notion that exon skip transcript is sufficient to induce a tail loss phenotype. Also, the news article claims that the study has highlighted more than 30 genes engaging in creating tails in various animals. This is correct, as stated in the original article, where they said, we screened through 31 human genes and their primate orthologs involved in tail development to identify a genetic variation associated with the loss of the tail in hominoids. Also, according to a New York Times article, the scientists ultimately uncovered an abnormality in the specific gene. TBXT is carried by apes and humans, but not monkeys. This was also stated in the primary article by scientists when they explained how a hominoid-specific ALU element was identified in the intron 6 of the TBX gene, which was unforeseen. They said, entailed species like a mouse, which has a heterozygous abnormalities in the coding areas of the TBXT orthologs, result in the loss or decreased shape of the tail. To be continued. The news article clarified that to evaluate the suggestion that genetic defect was implicated in the extinction of our tail, the scientists genetically manipulated mice with a TBXT genetic defect held by humans. This is true, as the primary article stated that they validated the existence of TBXT exon 6 transcripts in humans and its corresponding absence in mouse. Also, New York Times took the initiative to point out that according to the scientists, the TBXT alteration isn't the only cause humans have coccyxes rather than a tail. The primary article said it's possible that a wide range of tail qualities evolved in the ancient humans before ALU-Y was introduced. Even though ALU-Y insertion may have sparked a human tail loss evolution, subsequently genetic modification in early humans may have sustained the no-tail attribute. Lastly, the news article concluded that lacking a tail might have resulted in further complications. And this corresponds to the initial article's conclusion, which stated that they presumed the selection benefit must have been quite significant because the disappearance of the tail may have involved an evolutionary trade-off of neural tube anomalies, as demonstrated by the presence of spinal cord malformations. Overall, The New York Times article highlighted the key components and appropriately stated the purpose of the scientific experiment and their findings, even though it did not go into excessive depth on the findings in the primary article. This is my works cited page. The first one corresponds with the primary article, second is with a news article, and then we have figure 1, figure 2, figure 3, and figure 4. Thank you for taking the time to listen to my presentation.